Yeah, okay. It's really a pleasure to be here. And yeah, I think it's exciting to be talking about community ecology, its interface with disease ecology. How do we try and go about studying these communities that are of interest? And you would have heard a lot about this in terms of, thinking about community ecology from Dr. Umesh Srinivasan just before. So what I will do is get into the meat of what we are trying to study. So you've learned about community ecology. Now let's talk about what are the things that we are really trying to study here. So just for a purpose of repetition more than anything else, or reiteration, I should say, I'm just like talking about a definition of what a community. It's a group of species commonly found together. It could be plants or animals with similar habitat requirements, may influence one another in some ways, and may depend on similar environmental processes. So it could be, an assemblage of different mammal species, like you see this picture in Kaziranga National Park, or it could be, another mammals assemblage in Masai Mara in Kenya, and you could have potentially a community of bird species. These are water birds that are using this habitat. So there are different kinds of species that together comprise a community, and they could be influencing each other in different ways, and depending on similar environmental processes, similar habitats, and all of that kind of play out as we look at them further. So a few things to keep in mind. A lot of this applies to populations of animals, but also could potentially be relevant to when we're thinking about communities as well. So one thing is about how individuals or species space themselves, how they like distribute themselves in space. And there are different kinds of ways in which different patterns that we see in nature when we are thinking about different individuals of a population, or we're thinking about different species of a community. So one type of spatial aggregation or dispersion is what we call clumped distribution, where the animals are clumped together. So like how you see here, you could see these kind of distributions. And where does this happen? Typically, if you have gregarious species like elephants that tend to be social and tend to come together, or if you have highly clumped resources. So the resources that a particular set of species or a set of individuals in a population are looking for are very clumped. So then automatically you would expect that the animals will also be, spacing themselves around those resources. So for example, amphibians in deciduous forests might be clumped towards water bodies, streams, other such areas that are not evenly distributed throughout the habitat, but in certain places. Another type of distribution or dispersion that we see is what is called uniform distribution. And this is usually in the case of when there's a lot of intraspecific competition. What do I mean by intraspecific competition? When two individuals of the same species are competing with each other. We'll talk about it a little bit more, and which can lead to territoriality. So if you think about something like this, penguins in Antarctica. If you just look at this picture, you'll see that it's quite similar to what you see here. So they are very uniformly distributed. Penguins are extremely territorial, and they have their own little space that is their own, and then they don't want another individual coming into that space. So then they all very evenly space themselves out. So that's the kind of other kinds of territorial species also might show this kind of a distribution pattern. Finally, you could have a spatial pattern which is more random. This basically indicates a lack of strong attraction or repulsion among individuals. Like when we talked about territoriality before, then we're talking about clearly there's a strong repulsion, right? Or even like trying to hold on to resources. Here, you don't see any such strong patterns. And then you could have a situation where you have different species that are randomly distributed. If you take a rainforest like the Amazon or some areas in the Western Ghats, you might find a whole set of different plant species that are randomly distributed with respect to each other. This is just so you keep in mind in terms of how animals or plants of a community, how they space themselves out, will have implications for trying to understand the community as we move forward. So a few things that are playing out here in terms of influencing where different individuals of a population or where different species are present is in terms of the interactions that they have with one another. So you can have different kinds of species interactions. You can have competition for within the same species, so intraspecific competition. You could have competition between species, that's interspecific competition. You could have predation, where a particular predator then, like has an influence on its prey species. So all of these kind of have an influence. If there are two species that you're considering, you can have a number of different ways in which their interactions and relationships can play out. So you could consider a case where there's a positive relationship between the two. So what is referred to as mutualism. On the other hand, predation, parasitism, herbivory are all cases where one species is benefiting while the other species is negatively impacted, right? And competition is an example where both species are negatively influenced because of the interactions that they are having. All right, so who can tell me an example of mutualism? Any ideas? Yeah? Lichens, okay. Do you want to expand on that?
Algae and fungus, they together they live. So where algae provides kind of shelter to the fungus, and fungus will what it does, it absorbs the nutrients, it makes it more simplified, which is quite easy for the algae to uptake and do that. So that is a kind of mutualism.
Yeah, absolutely. Thank you. You had an example as well. Same example. Okay, let me see what examples I have here. Who knows about this? Yeah, you want to? Yeah. Do you want to use the mic?
So aphids starts to get not nectar, it's liquid from the plants. So they also release some sweet liquid for the ants, so they can come and protect them from the other predators.
Okay. So the ant is benefiting because of the nectar, and the aphid is benefiting because of the protection from the ants. Yeah, perfect. There are specialized pollinators, fig wasps. Everybody in Bangalore must know about them at least, right? There are also seed dispersers, like hornbills, dispersing seeds of particular plants. This one was considered a mutualistic relationship in the past because these are oxpeckers, and this is a Cape buffalo in Africa. And so initially it was thought that these oxpeckers basically pick off the insects on the Cape buffalo, and so it is positively influencing the Cape buffalo. But now more recent studies suggest that actually when they're picking off the insects, they're causing injuries on the Cape buffalo, and there's no significant positive outcome for the Cape buffaloes because of the oxpeckers. So I've just kept it there because sometimes what we may earlier think about a particular type of relationship may eventually, with more data and more understanding of a system, we might have a different view on it. Right? Commensalism is a case where one species is positively is basically positively influenced by the other. The other species doesn't have any doesn't experience any positive or negative impact. So a couple of examples are when you have lichen on tree species. Actually, the tree is not really benefiting anyway, but the lichen is. Or barnacles on whales. Again, the barnacles are hitchhiking across large distances. They are benefiting, but the whale really doesn't really make a difference to the whale whether it has barnacles on its tail or not. Then, of course, we have the ones where one species is positively influenced while the other one is negatively impacted. You have cases of predation, parasitism, herbivory. These are all different kinds of interspecies interactions, which will all effectively influence what the community is looking like and how it's going to change in time and all of those kind of things. Yeah? So coming back to what we are trying to study then, given all of this background that you have, this is what a community is, this is how different species may influence each other, how they're dependent on similar environmental processes. So if you think about what we are trying to study, these are the kinds of things we are really interested in. Which species make up the community? So effectively, we're talking about richness and diversity. Can somebody tell me what species richness is? What is species richness?
Number of species found in common terms.
Yeah, as simple as that. What about diversity? It's also like how number of species that are found, but the other thing that's also going in there is the abundance of the species. Yeah. So if you're talking about what is the makeup of a community, then we are looking at things like species richness and diversity. These are the things that we are trying to measure, understand, quantify, and understand, right? Then we may be interested in where they are found. So distribution of species. And this may be related to the environment, what kind of habitats are there and how that might be influencing it. It could also be related to different species interacting with each other. Just now we were talking about different kinds of relationships. So species interactions can also influence distribution of species. And finally, these may be things that we're thinking about in a particular snapshot in time. So we are really looking at spatial patterns. But if we were to continue to assess this over time, then we are effectively looking at how richness, how diversity, how distribution of species is changing in time. So effectively, we're looking at the dynamics of that community, different facets of that community, how that's changing in time, and what might be influencing that. So these are really the parameters that we are interested in: richness, diversity, distribution, the interactive effect of one species on the other, positive, negative, whatever it is, the kinds of different things that we would look at when you're thinking about community dynamics. These are the kinds of parameters that we will get into more in this particular lecture, okay? So let's consider a system here, all right? Let's say it's a particular habitat. So, and we are now thinking about it in time. So this is time t. This is t plus 1, let's say 1 year later, 2 years later, 3 years later, all right? Now here, we can basically have, if we are thinking about our first parameter that I talked about, species richness. Let's denote that by R. Okay, so R sub t is at time t, and then t plus 1, t plus 2, and t plus 3, right? So this is species richness over time for the same system. Now, what do you think is going to influence RT plus 1 compared to RT? Why would it change compared to RT, and why would RT plus 2 change compared to RT plus 1? What might bring about a change in time?
Environment it supports for its as well as environmental factors supports for its survival.
Correct, correct.
Environment.
Correct. Correct. So the key term that you are talking about here is survival. So survival of those species. If you have 20 species, if you have no change between t and t plus 1, that means all 20 species have to continue to survive to t plus 1, right? If a couple of those species die, and survival here doesn't necessarily only encompass the process of dying. It could also be that the animals or that particular species emigrates out of there, so moves out of there. So in either case, it's effectively like dying, right? So if you had 20, if 2 had moved out, then it becomes 18. So clearly there's a process of subtraction. So subtraction is one thing which can bring about a change. What's the other opposite obvious thing? Addition. How can additions happen? Migration into that particular system. You had something that you wanted to say?
Hello.
Yeah.
So RT is species richness, and that is like a binary vector. Like you consider species present, then it's 1. Species not present, it's 0.
No, richness is like a count, right? You're saying how many species are there?
Like a number, like
It's a
20
number, yes.
natural numbers, so
Yes.
0 to n.
Yes.
Okay. But then how do you differentiate between, let's say, a species going out and a species coming in, but then still then your RT remains the same? Although there's a dynamic, like, one species went out, another species came in.
Correct. That's a good question. So your richness may not change. You may have 20 in RT and you may have 20 in RT plus 1, but maybe 2 left, but 2 others came in.
Correct.
Right? So somewhere if you have the identities of the species that you're looking at. So you're looking at the composition of a particular community. So you're looking at a measure which is richness, but you also have information about each of the species. So then you will, I'll get to it in some time, but you will have an idea about how the composition is also changing. So yeah, so anyway, but basic processes, what we refer to, so this is your state variable, okay? Just keep that in mind. A state variable is a variable that defines the state of a system. There are X number of species in this system. There are X number of individuals in this population. That's population size or abundance. That's a state variable. This is species richness. It's also a state variable. And there are underlying vital rates or demographic processes that bring about a change in that community in this case, or it could have been also a population. So those vital rates we talked about, that survival, emigration, and we talked about immigration into the population, are what is bringing about a change in these state variables. Is everybody clear on that? Yeah? So the two vital rates that we are concerned with when we're thinking about this is gamma represents colonization. So new species coming into the system that were not there previously. And this epsilon here represents extinction, or species that were there in a particular time step, not there in a subsequent time step. Yeah? Okay. Now, instead of richness, if we look at distribution of species, now here psi, the symbol psi represents distribution. Again, you have distribution T, T plus 1, T plus 2, T plus 3. And what would bring about a change in distribution of species? I mean, effectively the extinction process could mean death. It could also represent emigration out of the, migration out of the population. Immigration is what would effectively represent colonization, because it's animals coming in to that population. So distribution, what are the things that might bring about a change in the distribution? What would be the vital rates?
Availability of food source.
Sure, but what are the demographic processes? We talked about vital rates. So what would be the vital rates? Sorry? What are the vital rates? So we talked about colonization and extinction for species richness. For distribution, do you think something different would be influencing it, or same, or what?
Maybe you can just explain what are vital rates, very briefly.
Yeah, so like I said, vital rates are the processes that bring about a change in your state variable, which is what is defining the state. The state variable previously we were talking about was species richness, saying how many species are present in this system, right? That is, what is the state of the system? There are X number of species in the system, right? Now the state variable is distribution. These many, this proportion of a particular area is occupied by species 1 or species 2 or so on, right? So that's your state variable, and vital rates will be processes, demographic processes, that will bring about a change in that state variable.
So the system is heterogeneous or homogeneous then?
These are all good questions that will influence maybe the vital rates in some ways to bring about a change, but suppose we consider for now a homogeneous system. Yeah.
What did she say?
I said it does matter, but over here.
Here it doesn't matter.
Okay, so for example, say something like birth rates. Okay, do you think birth rates would impact, distribution? Differential birth rates. Due to competition between? No, we're talking about a species.
Let's say there's one species.
Yeah, one species.
Okay, one species, and the system is, let's say, the size of the home range of that species. Either now, earlier, somebody had asked, what is species richness? Is it a binary? I said it's a count, right, of the number of species that are there. Let me say here, this is a binary variable. It's either 0 or 1. Either the species is present or not present. So 0 or 1 in a particular system. So what do you think will influence a 0 becoming a 1 and a 1 becoming a 0?
The same vital rates of the overall community.
Same vital rates. Same vital rates will influence distribution as well, right? A particular system or particular site, particular location that was previously occupied, if it's unoccupied, it's because of local extinction. It's either because the animal has died or because it has emigrated out. Same way, something that is unoccupied at a particular point in time, if it gets occupied in a future point in time, why will it happen? Because it's getting colonized by the species. It's moving in from somewhere else, and that is potentially what is influencing that.
This is an important point. You guys understood, right? Though you're talking about different things, richness versus distribution, it's the same processes which are actually driving those state variables.
It's just that what the state variables are different, and we're measuring them differently. One is a counter species, one is whether a particular species is present or not.
I have a point. If the system is homogeneous system where the single species were there, so if there is abundance of the single species, there will be a competition between them.
Yes.
If it means the resources should be much more enough to survive but particular species. Or else if the system is heterogeneous, it will be balanced because one species will be interacting with the other, it may be any interactions, it may be anything interaction, so it may be balanced. So we can say that only. You can correlate with these things, right?
Yeah, absolutely. So what would you say then would be the next logical thing to think about? Resources, what else? What might, see, finally, if this is your system, right, and these are your state variables, these state variables are changing because of two vital rates, colonization and extinction. Now, colonization and extinction itself will be influenced by so many different things. Resources may be one, species interactions and competition related to resources may be another. What about humans? Do they have an influence in all of this or no? Climatic factors may be another. Land use change.
Yeah, so humans, right?
Yeah, there are so many different things that can bring about a change, right? So these are some of the things, perfect direction in which, I mean, this is logically clearly flowing from what I've been saying. So first of all, there's population size of that species in a location will also influence it. If there are only two individuals, very high probability of them dying out and that place getting locally extinct. Resource availability, competition, predation, diseases and parasites. This is a disease ecology workshop. So it's going to be, again, one more factor that's going to influence this. Dispersal and migration out of there, right? Stochastic processes, catastrophic events, a tsunami in some place. All of these are going to influence things. And then, of course, we cannot forget that anthropogenic influence and disturbance has a very strong role in all of this. So you can have habitat loss and fragmentation, you can have hunting, you can have climate change, you can have invasive species being introduced to a system. There are so many different factors that can bring about a change in your system. It can bring about a change in your state variable, whether it's species richness or, what is the other one? Distribution. But how does it do it? It does it by all of these things basically influence the underlying vital rates in some way. Either it's influencing colonization rates or extinction rates or both. Yeah.
So, like, it's a basic doubt, even a species will be there will be invasion of new species. So, there is any different terms between invasion and colonization, or both are very different?
Yeah, no, that's a good question. So invasion, or when we say invasive, when we use the term invasive species, we're usually talking about species that are not native to that environment, that are somehow make their way to that environment, usually helped by us. But once they make their way to that environment, they don't have the natural controls that keep their populations in check in their native habitat, and therefore they kind of grow exponentially often. So they expand and they take over the place and they outcompete a lot of other species. But effectively, it is like colonization, but once colonization happens, that particular species is a much better competitor compared to other species that are there, and therefore it does much better than others. So that's, and it's not native to there. So there has to be some process by which, usually human mediated, that would bring it to that environment. Because if you're talking about colonization normally, it would have to be from a pool of species that are found in that larger area that they can colonize from. Once a particular invasive species is introduced in a place, lantana has now came as a potted plant to India, and now it's gone and proliferated everywhere. Now that lantana has established itself in so many habitats, you can imagine a scenario where colonization of a new habitat can happen by lantana, because birds might disperse their seeds or other reasons.
Great.
Yeah.
So I was wondering, I could understand, drivers of change. There are n number of, stuff that can think about. But when you're talking about communities, community ecology, so are there just two state variables that you all define for across, all the different, the two vital rates that you define across all state variables? Or is there other vital rates as well, apart from extinction and colonization?
Can you think of other vital rates?
No, I couldn't.
So if we are talking
Input, output, right? Like anything, like if you think of water, I mean anything.
biotic factors will influence the vital rates. See, just understand that there are certain external factors that are influencing the system through the vital rates. Whatever change comes about happens through vital rates. Either there are more deaths, more births, more emigrants, whatever. But those things are being influenced by these things. So these things directly don't bring about a change in species richness. These things directly don't bring about a change in the distribution of species. They bring about that change because they are influencing the underlying vital rates. And for that, it's always addition, deletion. Basically, those are the basic processes that are happening in the background everywhere. We want to understand, if you want to really understand how, let's say, disease influences the composition of a particular community or the richness of a particular community, then what we would want to understand is how does disease influence the vital rates, the colonization process and the extinction process. Only then will we be able to have a mechanistic understanding of, okay, so this is what disease is doing. It's bringing down the survival rates of this particular set of species, and therefore the community is changing in this way.
Yes, yeah? I think the question was, are there any other ways in which you might want to, so what is this? We're basically describing the system. And we're describing system change, right? So if you're describing the system as species richness, then your vital rates are the way in which that is changing is through extinction and colonization. But there are other ways also, to get back to your question, to describe the system. You might think about diversity or dominance or evenness. You might think about functional diversity or functionality. What are the functional traits? You might think, oh, I'm only going to look at forest species and what is the species richness with respect to that. So you can have those specifications. You might think about species composition, how does that change? So those are also things that you might think about in terms of describing the system. But I guess the most fundamental way to think about describing the system is just how many species are there, and that's how it changes. And once you have that fundamental understanding, everything else can follow from that same.
Those are layers that you can add on to further understand, describe a system. Complexity, nuance, all of that. But this is the fundamental, building blocks of trying to understand your community.
Yeah, and without that, you can't describe anything else. So it's basically the starting point.
Yes.
Yeah. One section.
My question is, how is competition helping the environment? Like none of the species is benefiting, right? Neither the species A or neither the species B. So what effect does it have on the environment or diversity?
So let's say, there are different, that's getting into a different, I won't go into details, but competition itself can play out in different ways. You can have a case where one species is a very strong competitor over the other. Let's say we were talking about lantana earlier. It outcompetes the other species to the extent that it drives that species extinct. So it's gone, and this species then just takes over that entire environment, right? You could have, as compared to that, you could have a scenario where the two species are competing with one another, but they're kind of at this nice balance where they are influencing each other, but they both remain, they coexist. in that environment. So depending on the kinds of interactions, the magnitude of that interaction, the magnitude of the negative impact, it will influence what happens down the line in terms of the environment. If one species is a very strong competitor for another, with another species, and that species is, let's say, an obligate, it's basically a specialized predator for a third herbivore species, then because that species is going down, that herbivore population may go up, just for example.
Thank you.
Yeah.
Great. One more question? Yeah, quickly.
So, competition, predation, these are interactions inside, right? So the drivers of change or vital rates are actually a function of the RT or SIT, right? So, but we are not taking that into consideration.
So actually, if you look at it, competition and predation are aspects of population of species. Like so, multiple species together make up the community. But if you just take two species out of that and then look at their interactions, that's when you can define or describe predation or competition more easily. And in that case, actually, the population size of each of the species and the interactive effect that they have with one another comes into play. So I'm not getting into those details here because then I'd be giving you a population ecology talk instead of a community ecology. But keep in mind that in community ecology, like when we're thinking about a community, there are many more such species, and maybe many more species are interacting with one another. So the size of each of their populations are going to have a role in all of this. But it's for the time being, this much is understanding it in this way is sufficient for us to delve deeper into the how do we study communities and what are we trying to quantify. Okay, so by now describing or talking about these drivers of change, we effectively go from what are we trying to study to the why questions. So we said which species make up the community, that species richness and diversity. We can then ask a why question. Why does species richness or diversity vary in space and time? Why does it change if you look at a particular environment? Why is it variable in space? Why is it changing in time? All of this is going to go back to some of these things influencing those vital rates, and therefore you see this change, right? Similarly, if you're saying the species distribution, again you can ask, why are species found where they are? Is it something to do with the environment? Is it something to do with how they're interacting with another species? Is it something to do with, I don't know, other climate change factors? Or what is it? So that's again something that we can ask, and that's where you really start understanding the system. Again, you can try and understand how species interact with one another. Why is one species reducing? Maybe it's because of the strong competitive effect of another species. And how things are changing in time. Again, you can ask many why questions related to that. Yeah. So before we get into that in more detail, first we should now what we thought is that I'll give you a kind of an introduction to the kinds of techniques we may use to try and sample our community, right? So we've thought about what are the things that we are trying to study, and we talked about why particular things might be influencing what we see. Now, how do we go and collect the kinds of data we require to be able to ask all these exciting, interesting questions, right? So there are different kinds of ways in which we can sample a community. It depends on the type of species that we're looking at. Let's consider the first one, which is what is referred to as point counts. Okay, now point counts is an approach that you can use to count animals of species that you can potentially, suppose I come into a particular environment, a particular forest or a habitat of a particular species. I could stand in one location and I can say, oh, 1, 2, 3, 4, 5, whatever. I can count a set of different species. So point counts are usually used in cases where you can have a sampling design, and Dr. Divya Vasudev will talk more about sampling design, so I'm not getting into it here. But just to say that you have a way in which you distribute a set of points in your area of interest. So if there's a particular area that you want to collect data from, you have some way in which you put some points there. These points are the places where you will go, and then you will count let's say in this case birds, you'll count the birds that you see around you, right? And what do we have to keep in mind here? Do you think you will be able to count birds as easily from this area as from this area? Yes or no? Why would that be the case? Yeah, absolutely. So then there is a possibility that you might miss some animals. So two important things to keep in mind when you're doing point counts is that there is a relationship with distance. The further the animal is, the higher the chance that you'll miss that animal, right? You won't be able to see. I mean, there's only a limit to how much you'll be able to see. You define a distance, maximum distance within which you're doing your counts. So let's say from this point, you're counting up to 100 meters. But even in this 0 to 100 meters, the probability of detecting an animal will go down as distance increases. There'll be more cover, there'll be more reasons why you will not see them. So one is a distance relationship. The other is to keep in mind that, let's say the yellow ones are the birds that you've detected from these points. So this one is 1 point, this is another point, this is another point. And the white ones are the ones that you've missed. So clearly you're not seeing every single species that is there. So you are detecting, like a proportion of the actual number of species that are there. So you're missing certain species, and you have to take that into account and quantify it, because otherwise what will happen, you'll undercount the number of species that are there, right? So detectability is the other factor that one has to keep in mind. So, but if you use a point count approach, this is a recently published work from our PhD student, Byung Same, who basically looked at this kind of a mosaic of wooded areas and agriculture in Meghalaya, in Northeast India. And he did point counts, and I won't get into the details, but basically a whole set of different bird species of different kinds of certain nectarivores, some granivores, some that depend on, like insects and so on, insectivores. And he's basically looked at their distribution in space and how that's changing. And he was able to do all of that by doing a systematic survey where he used point counts to count all the different bird species that he saw. Yeah. You can have non-invasive ways of doing point counts as well. So you could be the observer, you can stand at a particular location and you can say 1, 2, 3, 4. In certain other cases, particularly for species that are vocal, you can hear them, they call a lot, so it's how do we use that to our advantage? You can use acoustic recorders. You can effectively put acoustic recorders up in locations, they effectively represent the same thing as you standing at a particular point and counting. Instead of you counting, the acoustic recorder is recording all the calls. All the calls is getting from a distance. Again, same relationship, further the animal is, the call is going to be fainter and fainter, and so the acoustic recorder after a point is not going to be able to detect it. So there is again a distance relationship, but effectively the acoustic recorder is doing what you would do in terms of counting birds or counting other animals with a good vocalization. And here's an example of a paper that was recently published that has done just that. Yeah, there were a couple of questions.
So, for how long does this acoustic recorder function for? Because if it functions for, let's say, 30 minutes or 30 seconds even, a bird gives out a call from location A, flies to another location and gives out another call. Double counting, right?
Yeah.
So how do you take care of that?
Yeah, no, thanks. That's a good question. That gets into actually the sampling design aspects. Right now, I'm just telling you about the different approaches you can use in terms of monitoring these things. But yeah, there are experts here who can tell you more about it. But also there is, Divya will talk about sampling design related to all of these things. And in that particular session, maybe once the sampling design is discussed. So it really will be determined by the system you're trying to understand. So if you're talking about birds, what are going to be, considered to be independent counts, you have to decide and define, say, okay, I'm going to consider every half an hour to be a set of calls, then you keep a gap and then you again have another set. I don't know, Robin might want to say something more about this, but Yeah, so but I mean, it's a good question. We'll park it for now because I'm not getting into the design. How do you design a study like this? I'm just telling you about the approaches or the methods that you can use in terms of collecting the data. We'll get into the design after this, and then maybe it'll be good to discuss it then. Yeah?
So can it actually differentiate the vocals from a same individual?
Oh, yeah, that's an interesting question. We are actually trying to get at that with, so we are using acoustic recorders for different, like we're using it for gibbons. You know what hulok gibbons are? It's India's only ape species. They have a very characteristic call. I will not try to mimic it. But what we are trying to do with our acoustic recorders is to see, I mean, the human ear cannot differentiate the call of one individual from another. It all sounds like the same huku call, but we are trying to figure out if the computer is better than the human ear in that sense of looking at the signature of the calls and seeing if there's actually some difference between individuals.
Yes, just to add that very recently there's a paper on elephants, right, from Africa, where they're actually able to identify individuals. That's very cool. So this is the new frontier in terms of,
Absolutely.
I don't know,
Absolutely, absolutely. In fact, they've also like gone ahead and said that elephants are now, like the results have shown, or the data have shown that they are able to have like such fine scale communication with one another that you can effectively say that they're having a conversation. They have different names for different individuals in the herd, and they're like calling out to each individual. So it's really, a lot of things are getting opened up, and a lot of exciting, avenues of research and insights that are coming in through all of these new advances in technology.
So adding to our question, see if the same bird is in 10 meters distance, and if the same, it gives a call, and the same bird travels again for 100 meters and gives a call, does it differentiate? Means how can you differentiate that things?
Yeah, no, that's a very good question. I guess I have to get into the design, and Dr. Robin will also, maybe you can answer first and I'll say what I want.
Quick, yeah, just very briefly, with every sampling, with every kind of method, there are going to be some limitations. So, and it also depends on the questions that we want to ask. So I'm sure some of this will get covered in the sampling methodology, but bioacoustics by itself, we are currently having a workshop at ISER Tirupati, just kind of targeted at the kinds of questions that you all are asking, which is for my study, what would be a good design? So, we help design projects and things like that. So it's a little bit different from the standard, but the general principles of sampling and asking questions for a specific, designing the study on a specific question, that still applies. Yeah. Yeah, so there are so many things that are there, actually, which is why I'm not even attempting to answer the question. So,
but I'll try and answer it very briefly, which is that I was avoiding the sampling design aspects because it's coming up, but let me just say this much, that the point counts are meant to be snapshots in time. Okay, so you're taking something very quickly. So you cannot have a point count that goes on for 2 hours and you just keep accumulating species. That is not, then it is going to have biases. You want to have it, let's say, for 5 minutes or 10 minutes. In that, let's say 10 minutes is appropriate for your system. Then if you've got the same species, if you've got it called more than once, you basically count it as just, that species has been recorded already. Yeah.
Based on the frequency, if it is a short period of time, means 5 minutes, based on the frequency, if it is, if it has higher frequency and lower frequency, if the same bird travels to different thing, or it may be the same thing because it will be loss of frequency, it will be low frequency. Are you getting it?
Yeah.
So there is 2 or 3 different types of birds or belonging to the same species. The frequency will vary from distance to distance.
Correct.
Right? So your point is very valid and can be correlated with the things.
Yeah. No, thank you. Actually, so the type of the theoretical framework that is used often for these kind of things is what is called distance sampling. And distance sampling basically works with the idea that your detection of any given species is going to decline with distance. So when you're at the point, you will have a, and it's also used for line transects. So if it's a line, you're at the line and you're counting animals, you'll have a probability of 1 of detect. So anything that's at the point, you will detect for sure, right? And with distance, it'll decline, but that shape of that decline may vary depending on the frequency of how you are detecting animals as distance increases. So there's a whole, I'm not getting into it here, but there's a whole body of work that looks at the frequencies of detections at different distance classes from your point. So let's say 0 to 10 meters, 10 to 20 meters, 20 to 30, and so on, till 100 meters. And you can look for, at each of those classes, what are the frequencies of calls you're getting, and then you'll then form a, like you look at the, based on the data, you'll have a curve as to how that decline is happening, and that will let you estimate the detection probability of the species. So, but that's getting into something else, but it's a good question, and I think, did you want to?
Your question might just be, is this forest noisy or not noisy? Because the forest with more animals, etc., will have more noise versus another. Then you're just looking at decibel level across time. You may be interested in how many species are there. Then you want to differentiate across species. You may be interested in how many individuals are there. Then you want to differentiate across individuals. So it's not a question of can this do this or can this not do that. It's a question of what is your question. That was a funny phrase. But the main issue is what is your question? Do you want to differentiate between that bird standing 10 meters from you and 100 meters from you? Or does it not matter? Do you want to say that there are 10 birds versus 2 birds, or does it not matter? And what technique you're using, what question you're asking, will require different forms of data. If, for instance, you're looking at species richness alone, what did we say species richness was? Number of different species. Did we say that there are 10 individuals of one species, 20 individuals of one species, 1 individual of one species? We didn't say, right? That is an issue with that is a lack of granularity, that is a lack of detail in that metric of species richness. But your metric of species richness only requires you to differentiate across species, not across individuals.
Yeah, everybody is clear on that, no? So depending on the question, with these same data, you can do multiple different types of analyses. Again, for you to be able to do that analysis, your sampling design also has to be right, which is something we will talk about in a little bit later. So again, I mean, these are things that we can hear also, but there are other animals, and this is particularly relevant when it comes to disease ecology, because bats are often an important host species for a lot of disease ecology work. And for bat studies as well, acoustic detection is one of the ways in which you can potentially try and understand the community of bats. You have, like I showed you the previous, this is a song meter 4, and this is a song meter 4, but specialized for bats. So it basically collects information at the ultrasonic frequencies that we cannot hear, but obviously the bats are emitting those sounds, and the recorder is able to collect that information. And maybe I can ask Aishanya to very briefly talk a little bit or say a little bit about, because he did his master's work trying to detect different bat species at caves in Meghalaya using recorders. So just a little bit about, like how you use this kind of an approach.
Yeah, so bats, as we know, they call at much higher frequencies than we can hear from the human range. So the idea is to use ultrasonic bat detectors, which can detect as well as record these calls at higher speeds, at higher frequencies. And then what these recorders are able to do is convert them into a spectrum or a range that we can hear. So essentially you can do it in, again, different ways. One of the most simplest ways is to slow it down and basically transform the higher frequency calls into the human range. And again, you can do it in very different ways.
One of the most common ways is to, if you can catch bats, individual bats, and then as you're releasing them, once you've caught the bats, you're releasing them because bats again will call, they have very different kinds of calls. They have stress calls, they'll have social calls, communication calls. What we are essentially interested in is the echolocation calls, the calls that they use for flying, for echolocation. And one of the most discrete ways of doing that is as you're releasing the bat, you take a call as they release. So as they're flying away, that call is detected. That has to be an echolocation call, and we are sure about that. What I did for my master's thesis was what we call a passive kind of recording, where I was placing ultrasound detectors at the mouth or the entrance of the cave. So we know that bats are mostly nocturnal, at least the insectivorous bats are nocturnal. So as they leave their roosts or as they leave the cave entrances at around dusk time, you can basically place these passive recorders at the entrance, and as they are flying out, as they're using their echolocation calls to fly out, you basically detect and record them. The downside to that is that you don't know which bats are flying, because bats, visually identifying bats is very difficult. And similarly for many other cryptic species, right? It's very difficult, even for rodents, for example. It's very difficult to establish or do in like species level or even genus level identification sometimes. So acoustic recordings are very good examples of non-invasive techniques where we may not be able to get very precise precise identification, but it gives a very good method to basically get an idea of what kind of assemblages are there. And different birds, for example, have different calls based on their different adaptations to their niches. Similarly, bats have very different calls. Different species, different genuses have very different shapes of their calls, very different frequencies sometimes at which they call, a very different kind of signatures, as we call it. So yeah, acoustic detection in bats is something that is very important. I mean, bats are a very good example of like getting into different, depending on your question, getting into individuals or species without actually handling them or whatever, potentially harming them.
Great. Thanks so much, Ashanya. All right. Other kinds of non-invasive sampling, if you're talking about now more terrestrial species, one of the most popular ways in which to do that is to use camera traps. These are again, basically they take photographs of the individuals, and these photographs are taken because the camera is triggered by the animal itself. So earlier we used to use what were basically, there used to be an infrared beam, and that infrared beam would be cut when the animal is crossing, and then the cameras would get triggered. Now you have passive camera traps that basically sense the heat. They are heat sensing camera traps, and they sense the heat of the animal that's close by, and then when it does, it again triggers the camera. So it's opened up a world of possibilities in terms of trying to understand a lot of these species that are very elusive, low density usually, and difficult to otherwise find. But thanks to camera traps in these and that too like in areas that are difficult to sample, remote forests where it's difficult to look for these kind of animals, camera traps have opened up possibilities of looking at a whole set of different kinds of mammal species, ranging from large cats like tigers or leopards, jaguars and all that, to smaller carnivores like this is a golden cat that was taken in Nagaland as part of our work. But a number of different smaller animals, a lot of different kinds of mesocarnivores, even herbivores. You can look at communities of different species of mammals in particular using camera traps. So that's one thing. And with camera traps, there are going to be a few things to keep in mind very briefly. Where you place your camera trap will be important. It will depend on the type of species that you're trying to capture. So often trap locations, you need to put them in places where you'll have a higher, you're basically trying to maximize the photo capture of that animal. So you want to place it in locations, let's say a junction of two roads, usually if it's animals that are using roads or trails often to move, near water bodies if it's animals that are using water bodies frequently. So all of those kind of things have to be kept in mind. The height of the camera trap will also be important, whether you're trying to camera trap small animals or like animals the size of tigers. So again, there are a number of different things, design aspects that I'm not getting into, but essentially camera trapping is a really powerful tool to study mammal communities in many of these forests.
Captures both like pictures as well as sounds.
Pictures and sounds. Oh, that's a good question.
I was just thinking, in case there's a bird that doesn't call, but it's there, and you can see both, right? Okay, the bird is there as well as.
Yeah, I don't think I have come across any study that has used like a sound recorder with a camera trap together. See, the sound recorder is like it's on. It's just recording for the period that you keep it on. It's just recording whatever sounds are being emitted. This one is not on all the time. Otherwise, it will just keep taking pictures. Let's say these are the two camera traps here. Only when the animal here, human is simulating that. But only when an animal is crossing, it's going to trigger it. Otherwise, it would just keep taking pictures continuously. So it doesn't happen like that. But I don't know of, and actually, I'm not yet sure what kind of question you'd want to ask where you want the sound and the camera trap
But for
together.
example, with genetic data, that's always been like a dream to design a drone, which will go and like pull out a bit of tissue from an animal which is being camera trapped or something like that, right? Or for example, with rodents or bats, when you catch them, if there's some way to quickly collect samples, that would be really useful in the context of disease ecology. If you could also not just look at the community, but also get a sample. But I don't think there's any. Maybe in the future there'll be automated ways to do that.
Yeah, maybe.
Not right.
Well, one thing that is used, continuing from what Uma said, you are with genetic studies, you're trying to collect samples, genetic samples from, let's say, hair or scat, and we were going to talk about it in a moment. But you could combine that with camera traps because then you can also look at, this is the visual ID of this animal, and that has given me this genetic sample. And that could potentially allow certain kinds of questions to be asked, which may or may not be as feasible with just the genetic sample. So some combinations like that would be useful. Yeah.
So just wanted to share, there's this really interesting thing I saw. They use it with camera traps. They use AI for individual detection of animals.
Yeah.
I've read it for whales, I think, where it's not easy for them to identify individuals, but with the markings or the scars that they have, they use AI to detect them, and that's really interesting.
That's the holy grail for some of the work that I am doing, because we try and identify individual elephants based on their morphological features. So like with tigers or leopards, they have these, so tigers have stripe patterns that are unique to every individual. So it's much easier to have AI say, okay, this is a particular pattern that I see, and so this individual is different from another individual. With elephants, you use a combination of features, like you look at the ear morphology, you look at the tail, you look at the tusks if it's there, and only with that combination you're able to say. So we're working towards getting AI to be able to tell us to do that, but not as yet. But yeah, eventually if that happens, so it's great that it's happening with whales. I think whales is usually like scars on the fins or the tail that they're using. If they're able to automate that process and quickly get IDs of individuals, then that would be amazing. What I do know is that camera traps now are set up with, a lot of them are set up with, they're connected basically either through GSM or through satellite. They're connected to a network, and when a particular animal, so AI is able to not differentiate between individuals, but it can say, oh, this is an elephant walking, or this is a tiger walking. So moment it's detecting an animal like that, it sends it out to a remote location, and you're immediately in real time, you know that a particular type of species is moving through a certain area. So that is certainly happening as of now.
Yeah, just wanted to comment on the acoustic and camera trap.
Yeah.
I think there are two things here. I mean, it is technically possible to do these tandem deployments, not a single unit. But the question to why that would be necessary is the bigger thing. And usually it is because people don't believe one mode of data. And it's usually, there were times, if you think about the past, if you want to say that a species is found somewhere, you have to get the specimen and then match it to the type specimen in a museum. So that was the truth, right? And then later photos started getting accepted. And now we are getting to a time point where we are accepting sounds, but people may not believe it. So we work on Jordan's cursor, detecting it through acoustics, but people ask for photos. So we try and do some tandem deployments in that case. But you can imagine that sound travels much farther. But then there are, as you pointed out, some individuals, sometimes they may be quiet, but they may be moving in that area. So tandem deployments are possible. In birds now you have GPS tags which have audio recorders on them. But that is mostly to understand behavior, like when do they call and things like that. So these multiple types of data, it really depends on the question more than just the technology.
Yeah. Thanks. That's really helpful. Okay. Also, a few things, I guess.
So, excuse me. I have some doubt because some species which do mimicry of others, like for the production, like things. How can you differentiate that? But sounding birds.
I have to ask a bird ecologist to answer that. How do you differentiate between a racket-tailed drongo and the actual bird is trying to mimic?
Yeah, so this actually happened when we worked on the Jordan's cursor. We set up recorders for 8 months, listening every minute of all the nights. And then we found one call of the Jordan's cursor. So quantitatively, you can do analysis which says that it's the same. We sent it to a bunch of experts and they said, yes, this is Jordan's cursor. But there's one thing that is there, which is the context of the call. So it turned out that it was a shrike mimicking various birds. So if it was Jordan's cursor, it would just make the Jordan's cursor call repeatedly. But here it was making red-whiskered bulbul and something else and something else. So it's not just that signal, but the context of the signal that can help you determine mimicry. It may be tough sometimes, but most often the mimics are trying to increase variety. So there will be other things in succession.
Okay, just a couple of quick points here that, essentially if you have, let's say, a study area like this, which is green, this is a river on one side, and you could have home ranges of a particular territorial species like this, which are all the red polygons that you see here. So what you would want to do often is to try and have, you try and cover the home ranges of all the different individuals that might be there. Usually you'd place like 2 or 3 camera traps per home range of the individual. Depends on the question, and this is not getting into the details of study design too much, but depending on the questions, you would want to decide how you place your camera traps. But in general, that's the kind of frequency with which, or density with which you want to place your traps. Other kinds of ways in which you could study communities of animals, particularly those whose tracks you can easily see on a substrate, are what are called sign surveys. So you can have sign surveys for, different kinds of mammal species where you can look at their pug marks or their tracks, hoof marks, and other things, and then say, okay, this is this species, this is this species. So depending on the type of question, you're able to use such data also to ask, to understand the system better. You can also use their fecal matter. So you can have, elephant dung here. I was collecting this during my PhD fieldwork. But here, other kinds of, fecal droppings, deer, this is wild pigs. This is, I think, meant to be of otters. So depending on what species you're trying to look at and the study area that you're focusing on, you may have a whole set of different kinds of droppings of different animals, and then you can use that information to look at distribution, to look at other kinds of species composition, various kinds of questions that may be of interest when you're trying to understand the community. Yeah, and this is just an example of how we used such data to look at animal migrations between the floodplains of Kaziranga National Park, that's on the southern bank of the Brahmaputra River, and these highlands to the south, which is called Karbi Anglong. So just the movement of animals based on the distribution of their signs, basically tracks and their pellets. Yeah.
Apart from the scat samples, there is this figure of a cat rubbing. So.
Oh, sorry, I missed that. Yeah.
Can we, like, what are we doing? Collecting DNA samples?
Yeah. So I wanted to say, I forgot that. The other reason why, like, so with this, you're just looking at, okay, there are this species present, that species present. Here, in addition to saying that there were elephants here, you can, if it's fresh, you can also collect samples. I was collecting DNA samples here. I forgot, completely missed my mind. Thanks. So collecting DNA samples for other kinds of analyses and trying to understand other things. Here, this is basically, you have like these poles here, and you attract, you can use a particular scent to something to get cat species to come there, and then you have I think I might have had here. So you have something like this, right? They come and then they start rubbing, like cats being cats, they rub themselves and leave behind a lot of hair, which then you can use for various kinds of DNA analysis. So it's like a hair snag or
basically our life. That's what a lot of people in our lab do. We pick up all these hair.
Yeah, and other ways of getting at hair also. This is like a barbed wire, but often used for bears. If they're trying to cross through, then like a tuft of their hair will get like caught in those wires, and then again that hair is being used for DNA analysis. So if you're trying to sample such that you can collect DNA for these species, then you can rely on their dung or whatever fecal matter, but you can also try and get their hair samples. Uma, did you?
So wanted to add that, for example, in disease ecology studies, feces itself might be very important because many pathogens might be shed through the feces or urine, right? Or sometimes saliva. We actually do find sometimes lick marks and things like that. Those can potentially be important for primates. They may chew on fruit and throw it down. So you could use that. Hair per se may not be so useful in the context of disease ecology, but to look at the community, which species are where and so on, it's very useful.
Thanks.
One more question.
Yeah, there's a question there.
Has there been efforts to recover DNA from musk, like when the tigers, spray, yeah.
Spray is urine. Yeah. So people have done that, yeah. It works, yeah.
Okay, one last question and then I guess I have to move on.
So there is this GPS collars which we use to record the movement data of animals. So under which sampling technique does it actually come?
It's none of the things that I've talked about so far. Because, so with GPS collars, you are interested in looking at, like you said, movement of animals, of individuals in a population, how they are moving, how they're using space, what are their home ranges, all of those kind of things which are, slightly different from trying to understand the entire community. So then I didn't get into those methods here. Okay. But yeah, there's a lot of very interesting and important information that you get from using GPS collars. But yeah, I guess we won't talk about that just now. So from a disease ecology standpoint in particular, we want to also many times actually handle, so we were talking about non-invasive sampling so far, but many times we want to actually capture the animal and handle them because we want to collect samples which will allow us to understand various questions related to disease ecology. So first thing over here that you see is a Sherman trap that's used. You typically might bait it with peanut butter and other things, and then you get small mammals, rodents, that come into it and then get caught there, and then you're able to handle those animals and get samples from there. Very briefly, maybe Arjun can tell us a little bit more about that, because he just recently did some work on that for his master's, so yeah.
I mean, trapping small mammals is we need to get genetic data or samples out of them, handling and trapping is one of the main. I think we briefly spoke about this yesterday as well when we were talking about like how difficult it is to trap animals, and most of the time we end up putting 100 traps and get some 4 or 5 individuals caught in them. But then when you are directly trying to take samples, especially for disease ecology purposes, when you are taking, I mean, something that Ansil was also doing in Kardamane and Kudramukh that was explained yesterday. So I think it's you have different kind of traps. You have different size traps. Some which are also not the size, the sensitivity of the trap also differs. So you can have traps like set in such a way that only some body size animal would get caught. For example, those bigger traps in which you see the fox, there you have like bigger tomahawk traps and stuff where you have like even for larger small mammals like bandicoots and stuff, you can use them, and even for mongooses and things. But yeah, I mean, mostly it helps in doing, like, for example, land use, land cover sort of work, which I was doing, where I was deploying these traps in different land uses to see what the community structure of the small mammals are like, and also to retrieve samples and genetic data. Like I explained earlier, it's very difficult to get to the species identification of small mammals as well just by morphometrics. So you sometimes require genetic sampling for that. So
yeah, great, thanks. So then he's covered tomahawk traps as well. That's for bigger animals. And then the last thing that you see over here are what are called harp traps. And these are, if you can make out over here, these are all bats. So they're bats that get like trapped here, and then again you have individual bats, and then you can collect samples from them. Like using mist nets for birds, maybe that was discussed earlier. So there are different ways in which you can basically trap animals such that you can collect their samples and then let them go. Yeah. Oh yeah, absolutely. It's called a trap shy response. So there are some animals that are trap happy and others that are trap shy. So during my PhD, we were like, I was assisting a professor who was using these tomahawk traps to capture raccoons and possums. And there were some raccoons that would repeatedly, so every day there would be, like some kind of bait that's put there, and then the animal would come for the bait and get caught. So there were certain individuals, and we knew these individuals because they were all chipped. There was a unique chip that was placed on that animal. There were some animals that would come there every single night, get caught, and then get handled and go back. They just know that, okay, these guys will just like make a fuss about like, oh, measuring my hand and my arm and whatnot, and then they let me go. So there's no problem. I get this amazing food, and then I can. So that's a trap happy response. There are certain other individuals who, once they were caught, they were never caught again, and that's called a trap shy response. So yeah, certainly there's a behavioral response to these traps for sure. Yeah. So I think Divya, I think I'll stop here, and then Divya will talk about the study design aspects, and then we will continue. So we've decided not to have me talking throughout, but so I'll come back again.
So while I'm opening this up, can I just know how many of you are from an ecology background? Some. Maths background, I think. Some. Statistics. Oh, no. I'm trying to figure out who it is. What background? But disease background. Anything else on this? Any other? No. Oh, okay. Still figuring it out. People, 1, 2, a couple. Okay, still figuring it out. How many of you have gone out into the forest and done some of these surveys? Like use camera traps, use recorders? Quite a few of you. So, can some of you just say as part of what? What about it?
Ansil.
You can just say loudly. Can you all hear me?
Do you know how to get out of this to the?
Is there a? I need to give me a mic.
Yeah.
So we are doing line transact for the estimating the frogs population.
You're doing line transact?
Yeah.
Okay. Did you choose the line transact?
Yes. Previous PhD students he chose. So it's a long-term study.
Okay, okay. So you were just given the points and you went over there.
It's not point. It's a line transaction. Yes. So every year we need to go and it's collecting that. Yeah.
Who else had their points? You had your hand raised.
Can we open a different presentation?
Yeah. Pass the mic over.
Oh.
I myself didn't do bat recording, but I assisted in bat recording. Yeah. So I work on bats.
You currently work on bats?
Yes, yes. I currently work on. Yeah.
You did bat recording?
Yeah. I'm student at Rumas. Yeah.
So you went out and recorded?
Yes, I have done.
Where did you go?
Near Kolar here.
How did you choose where to take your recordings?
So basically, where they are out for foraging and in the inside the bat roost.
Inside the bat.
Yeah.
So you knew where the
Bat roosts are there.
and you purposely went to that area?
Yes.
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 Not everybody works in the forest. Some people might be working in the urban settings. Sure.
So mine was a rodent trapping. So we basically go to the selected villages in the households.
In whose house?
Villages, households.
Okay, villages,
Yeah.
villages. All right. Go ahead. What is that? Oh, thanks. It's got a clip, no? I can just put it on here. Thank you. You can all hear me? Yeah. Okay, I don't have to shout anymore. So you did rodent trapping in near people's houses?
Yeah.
Okay.
Surrounding the houses.
Surrounding people's houses.
Paddy field, basically. Yeah.
And what was the aim? Like, why did you do it near people's houses?
To detect presence of infection, to capture rodents
Okay.
for the infection studies.
Okay. And where did you place your traps?
Across, like where we could sense there could be a movement of rodents and shrews,
Okay.
like on the edges of the houses and the places where they store rice and stuff, and in the
Okay.
kitchen, something like we know like where they usually have their food,
Okay.
and then they move across. Yeah.
Okay, so I see two people basically who have know where the animals are and have gone there and placed their traps over there. I won't ask the ecologist, I guess. So, all right. So I'm going to talk a little bit about. It's going to cover some things that some of you might already know. Things like models, etc. Some of you might already know a little bit about, but it's worth still covering. And some part of how do you sample an area, any particular area which is of your interest, in a way that can help you answer whatever questions you have. So some of the questions that you guys asked fed into that, like how do you do some amount of sampling such that it answers every question that you have, any question that you have. These are just going to be basic principles, but we can circle back to those some specific questions that came up last time. All right. What is the point of doing all this? What is the point of like going out there, surveying, putting all of that effort, getting all of that data? There are a few things that we want to do. We want to quantify something. We want to quantify it because we want to measure it across time, across space, something. So we want to quantify something in our data, from our data. So it might be species richness, like Varun was talking about. It might be what proportion of the area is covered by a particular species. It might be how species richness, how many species there are. It might be how many specialist species there are, whatever it is. We might want to quantify something from our data. We want to have some sort of a metric from our data. We have some specific questions and we want some formal way, a structured way of answering those questions. We want to make sure that our data and our inference are particularly answering those questions. And we not only want to make inference about that one rat that we've captured or the one bat that we have found, we want to make some inference about the larger population, correct? We want to say something about the larger population of rats or larger population of people in the area who are exposed to rats, or want to make some inference about the larger population of bats. Same thing with the GPS surveys. We want to make some inference about the larger population of animals that are moving. In light of uncertainty. So we don't know everything about the system. There's lots of variation. In light of that lots of variation, we want to make some inference. But importantly, and I want to reiterate this, we don't want to miss the ecological forest for the statistical trees. So the aim is not to do the statistics. The aim is to answer the ecological question. And we need to make sure that our sampling techniques and our statistical techniques help us answer the ecological question. Which is why throughout the last 1 hour or so, every time, many times when there were questions asked, the answer was just, what is your question, right? So that is very important, has to always be there in your mind, what is your question? Which is why also some methods, for instance, like GPS collars don't get covered in this, because that is not a technique which is appropriate for this particular question. All right, so, what is you guys have all heard, how many of you have dealt with models? All the mathematicians, statisticians would have dealt with models. How many of you have dealt with models? Many of you. What is a model?
Something which captures the dynamics or something which captures the essence of a system.
Very nice. Something which captures the essence of a system, right? So a model is some representation of your system of interest, but it captures that particular aspect that you are most interested in and sort of like simplifies the rest of it. Yeah. So a model is a simplified form of your system, because often ecological systems are very complex systems, or any of these systems, it might be wherever it is, they're very complex systems. So we're trying to simplify the system, but really focus on what our aspect of interest is. So over here, if you're interested in elephant foraging ecology, this basically ignores like a little bit of the shape of the elephant maybe, but it really focuses on that elephant eating. Yeah. And this is really important thing to know, that all models are wrong, some models are useful. So we're looking for that model which is useful. Right? So the first step to a model might be a descriptive model, right? We might just want to describe. So it's a representation of our system. So over here our representation would be what? Elephants are eating grass. That's a representation of our system of interest. So descriptive models might be like really simple things. We might say human populations are increasing exponentially, tiger populations are determined by prey populations, hunting impacts Amur falcons, overfishing is bad. Are these good models? Are these very useful? Why are there's some shaking of heads? Nobody's very impressed with these models. Why are they not very, what could be done to make them more useful? That is okay, that is what is happening over there. Say, let's take one example. Tiger populations are determined by prey, or hunting, overfishing is bad. Let's take this one. Yeah, sure, overfishing is bad, but if we want to go out and test this model, if we want to go out and collect data for this model, if we want to go and measure something about this model, does it lead us to it? Kind of does. It's very vague. It's very vague. So we might want it to be a little bit more specific. Remember, everything that Varun was talking about in the last class was what are the metrics we can think about? What are the vital rates? What is the system state? It was very specific. So we can improve these models by making them a little bit more specific and making them measurable, right? So we talk about them in terms of measurable quantities. Human population size, we can measure human population size, right? Is increasing exponentially. That's a function, we can see whether it's increasing exponentially. Tiger population size is determined by prey population size. Both are things that we can measure. We can see, measure one, measure the other, see whether this is true or not. Hunting, we can say hunting rates maybe impacts survival rates or mortality rates. That's something again that we can measure. So a good model is something that uses measurable variables. And a first step to doing anything is always having a good model, right? Which arises, of course, from your question. From your what? Did somebody say PhD? Need, from your need, correct. From your need, from your interest, and from your question. Yeah. Overfishing leads to change in age structure or change in size structure of fish populations. Now that's something that we can measure. Directly leads us to our methods. Yes? All right. You can go one step further. You can become a little bit more fancy. You can start getting notations in place. So we have, let's focus on this. It's an easy one. All of us like tigers. Everybody doesn't like, anybody here doesn't like tigers? Umesh is raising his hand. Robin, the birders are like, no, thank you. Tiger population size is determined by prey population size, right? So we can have, a notation for this. We can say N of tigers, abundance of tigers, is determined by some form. We'll figure out the form later. By N of prey, by the population size of prey. We can make it simpler and say presence of tigers. So now we've come down to a binary. Are tigers there or not is determined by whether our prey is there or not. Yeah. Is this the only thing that might determine where tigers are? What else might determine? Huh? Habitat. Habitat might determine. Environment, you said the same thing. Habitat, everybody thinks that maybe habitat might determine where tigers are. So we might, that means have alternative hypothesis, or we might have, so alternative models, or we might have even models with increasing complexity. So we might say, yeah, tigers are determined by that, but also by how much forest there is in the surrounding area. Note again, these are measurable quantities. It may, this is an additive, this is an interactive model. Everybody knows what the difference is. Somebody who said yes, somebody said yes. I saw some people saying yes. As soon as I was like, somebody said yes, everybody is like. Some people said yes. Who knows the difference between an additive and an interactive model? Yes, go ahead.
So in additive model, prey density and forest cover independently would affect tiger density.
Okay, perfect. So that's the first half. I'm going to let some other voices also. So in an additive model, I'll repeat it for the recording. The prey and the number of prey and the percentage of forest are going to independently impact the number of tigers. Interactive model?
Interactive model tells about the prey, number of prey interacting with the forest. So these two variables will be interacting and how it collectively affects the population of the tiger.
How it collectively, so basically the impact of the number of prey on the number of tigers is going to depend on how much forest there is. If there is no forest, there can be 1001 prey, but there will be no tigers, for instance, as an example.
Hello. Actually, what did make the difference when interacting prey is added with the percentage of forest or interacting prey multiplied with the percentage of forest? What that makes difference? The second point and that.
This one?
Yes.
It means that the impact of the number of prey on the number of tigers will be different when there is less forest and when there is more forest.
Okay.
Suppose, for an example, if there is less forest, maybe the number of prey.
No, I'm actually, what is the plus sign and into the multiplication?
That's exactly
line.
what it is, an additive effect and an interactive.
Interactive, okay. Thank you.
Does it come down to independent and dependent variables?
No, it doesn't come down to independent and dependent variables. Independent and dependent variables was a term that was used earlier for this side, the left-hand side of the function and the right-hand side of the function. Nowadays, we broadly call it response variables and predictor variables. Or response variables, sometimes we'll use the term covariates. We might also use the term parameter, and I'll just come to that. One of the reasons for not calling it dependent and independent variables and calling it response and predictor variables is because a large amount of work that happens in the wild, like in these natural ecosystems, is observational. There is also experimental work that happens, but it's also observational, which means we can't control the system. And that means we have little bit less power to talk about causality.
Could you just continue the analogy?
Continue in the analogy?
Yeah, you were just telling us.
Independent versus?
I mean, what is independent? Additive. Yeah, additive.
Additive versus interactive? Yeah. Okay, so suppose you have very little forest, yeah? Then maybe the tiger is really dependent on the number of prey. So the relationship between number of tiger and number of prey is very steep, is very strong. Maybe when you have lots of forest, for some reason the tiger is not as dependent on the number of prey. So then the relationship between the number of prey and the number of tiger is a little weaker. It might still be positive, but it's a little weaker. For example, that is one example of an interactive versus an additive relationship. And you can have something else also coming into play. So are more complex models always better? No. One person for no. Are more complex models always better? Two people for no. Any yeses? Why are more complex models not always better?
Once again, it comes to the number of variables that you can control or you cannot control. In the complex models, there is a chance where you have n number of variables which you cannot control. But in the simpler one, so probably while
You can test.
Yeah.
You can test simpler models better. Right. You can, correct, you can test simpler models better. Going back to your simplification of the model, you can do that better. And you might be overfitting to, if you're thinking about, like if you're adding too many parameters in. So you may not have the power to test too many models, and you might be overfitting if you add too many parameters, right? And we'll come a little bit to that, but basically, again, just to clarify from dependent versus independent, for the purpose of for the next talk actually, these terms over here on this side of the relationship that we're really interested in, or your response variables, we'll refer to as parameters. These are the main things that we're thinking of estimating. So for instance, when Varun was talking about distribution, he spoke about occupancy as a state variable. That would be your parameter. When you plug it into your model and you calculate or you estimate what a value of that parameter is, it's called an estimate. It's called an estimated parameter or an estimate of the parameter. This estimate usually comes with some form of uncertainty. What you think is going to shape those variables, so what earlier might have been called independent variables or what are called predictor variables are in this modeling framework is also called covariates. So these are the covariates that you think might be influencing your parameter of interest. So these covariates can be various different things, and this basically forms your question of interest. Is forest impacting tigers? That's your question of interest. Right? So is forest your covariate impacting tigers your parameter? And the entire thing put together is called your model. Yes? Any confusion with this? Okay, so the question is, what is the best model, right? And it's very, very important. The reason that I'm starting with this is because it's extremely important to start off with an a priori model set. That is, before you go out, before you do your study, you need to know what is your question. You need to very clearly have measurable variables on both sides of your equation. And you need to say, these are the things that I'm interested in as my parameter of interest. These are the things I'm interested in as my covariate. Just to say, if you have any given phenomenon, there can be 1001 different explanations that you can come up with. Nowadays, with remote sensing, there can be 1001 different variables that you can come up with, but which is why it's really, really important to have your a priori model set and go in with a question in hand. Complex models are not always better. What we're looking for is the simplest models with the best fit to the data. How do you get the simplest model with the best fit to the data? Varun will talk about a little bit more later. So now, how do you then decide which is the best model? You essentially see we are talking about simplest model with the best fit to the data, right? So we can always say which is the simplest model over here. It's the model with the least parameters, but you need to also get at that best fit with the data. So we have to go out, get data, confront these models with the data, and select which is most likely. All right, the other thing before talking about how do we go out and get that data, that's the big question, no? Other thing before doing that I just want to talk about is what are called hierarchical models. For the rest of, that is in the afternoon session, what we're going to be talking about are hierarchical models. Yesterday also you might have dealt with these, but consider a system. There's a forest area, this is your landscape of interest, might be grassland also. There is some variation across the system, there's a water body over there, there's some cliffs and things like that, there's like some human habitation, etc. Is there deer in this area? You're interested in the deer. Is the deer equally distributed across space? It's kind of clumped in one area, not so much there in like this bottom area, right? So this is your system. There's some process that's happening over there. You might be interested in how many deer there are. You might be interested in where they are, what drives their presence. You might be interested in any of that. But this is your system. And there are various ecological processes that drive your system. So that's the ecological system or the ecological processes. You are going to go out there and get your data, some form or the other. You might be going only to the cliffs, you might be going only to the roads. So that's you on top of that. That's your observational process. You are doing something, going out there and sampling the system in a particular way, right? Together, these two things are going to lead to your data. So you have to, this is what you're interested in, the ecological process. This is what you get, the data. You cannot ignore this along the way and jump from here to here. You have to consider that ecological process as well, how you have sampled your system. So, how do you sample your system and what are the issues with sampling your system? And this is especially true, we're talking in like the context largely of largish, complexish, heterogeneous landscapes, which may have some forests, may have some grasslands, and so on and so forth. Some of it may be accessible, some of it may not be accessible. Issue is you can't go everywhere. You can't sample every animal. You cannot see everything. Some of you already asked these questions. If you put a recorder out for 10 minutes and a bird calls at the 11th minute, what do you do? If you have one animal which is calling next to your recorder and goes out and calls just away from your recorder, what do you do, right? So there are going to be issues. So you cannot see everything, you cannot go everywhere, you cannot sample every animal. You can't measure the entire population, and you don't know your system in entirety. For the most, you don't know your system in entirety. How do you sample in these contexts? Putting it into a little bit more less casual words, you can't go everywhere, which means you need to think of some sort of spatially informed survey design. You can't sample every animal, again fits within the realm of survey design. You can't see everything is what is called the detection process. Varun already referred to it a little bit. You already spoke about in terms of where can you see animals more and you can see animals less. Distance. You already spoke about it a little bit in terms of distance, but there is a detection process also that you need to consider. And then we'll talk a little bit about uncertainty. So for the rest of this hour, this is what we're going to be talking about. Break for lunch? No. Either way, is lunch at 1? Oh, it's supposed to be. So we can go till 1 and then we can. All right, so you can't go everywhere. Let's go back to our system. This is our system. We've got all of these things over there. You are over there. You've come just to the edge of the forest. And where do you sample? If you're going to place your camera traps, recorders, you're just going to take binoculars and go out, you're going to do point counts, where are you going to do it and how much of this area are you going to sample? So would you go there? You know that there are many animals there. Huh? No, lots of shaking of heads. Why not? There's so many deer over there. What else do you want? You're interested in the deer only. Very good. What is your question? Okay, very good. If you're interested in how humans are interacting with deer, then you'll study it somewhere over here. I think that question is also there somewhere. It's not there. All right. If you're interested in assessing maybe like how many animals are there in this entire forest. Huh? You want to go to multiple locations. Okay, you want to go to multiple locations. Which locations would you choose?
You can just put your cameras near water bodies because they will come anytime.
So they'll all come to the water bodies?
Suppose there is a single water body, then they must have to come sometime, right?
Okay, so if there is a single body, then if you want to count the number of animals and you go to the area where you think that many animals might come, all of them you can't control, right? Many animals might come. Is it a question for everybody in the audience? Is it okay to just go to the locations where you know animals are there more?
We can go where the animals will take rest, means where they hide, means in night times, where they settle, yes, where they settle. Actually, where they settle after reaching all the things and eating all the things, activities done with, and where they settle at night. So we can easily estimate the numbers, right? Rather than going with random things like going different locations like that, because set of clans or set of herds have particular locations where they settle at night, right?
You don't always know those locations in light of uncertainty. You don't know whether all the animals are going to come to one place, some more uncertainty.
Then we can divide that into different quadrants and we can estimate based on the density, right?
Divide the entire thing into
quadrants,
grid
different
cells
grids
or
and
something
cells,
like
and
that.
we can count down based on
Okay,
number
let's
cells.
keep that and let's think about how we would divide it into quadrants. Okay. So what happens if you go only to the locations where there are more animals? You might overestimate the population size. You'll have a biased estimate because this area is not representative of this area, right? So if you move from this area to this area, extrapolate from this area to this area, that's not going to be representative of the entire population. This is a common one. If there's a road going over here and you're like, who's going to walk all the way? There's so many leeches, ticks and all of that. No, don't want to like crawl through lantana and get to that water body. Is this okay? It's still not representative of the entire system. Now, there is an important thing over here. What did we say? In light of uncertainty, we actually don't know how many animals are there, how many animals are here, how many are here. So we don't know whether we are underestimating the system or whether we are overestimating the system. If we go over here, we'll find absolutely no deer. We'll be like, this place is useless, it has no deer. That's also not correct. No? Okay, so the question is, how much of the focal area do you sample? Where do you place your camera traps? Where do you sample? If you're putting quadrants, say you're going to, even if you're putting quadrants, you have to go to some location, right? Your transects, like the frog transects. Where do you put those transects? So that in entirety, how do you choose your sampling units is what is basically covered in survey design or study design. So the idea of study design or survey design is to say, okay, how do I choose my sample units, which may be locations, it may be animals, depending on what your questions are, from an entire population or from an entire location, such that whatever I estimate from that smaller sample can be extrapolated to the entire population. So you're going to go to just 3-4 places over here, but you want to make inference to the entire area. Why? Because you can't go everywhere. You can't see every animal. So this is really critical. Whatever locations you choose must be representative of the entire population. How do you make them representative of the entire population? Anybody knows? Yes.
Okay, so you have your grids throughout the region, but it's not possible to go to all the grids and you can't be biased, I'll go there and sample. So random sampling along with mark recapture.
So mark recapture is a separate part.
Yeah.
The first part is just how do you choose? Mark recapture is how you're going to analyze your data later. Your first question is where do you put your camera traps if you're talking about mark recapture or whatever method it is. So you're suggesting random sampling, right? So one suggestion for choosing randomly, one suggestion for choosing multiple points, not just going to one point, but choosing multiple points in your entire area. Both are correct. Basic 3 fundamental tenets that have been there for a really long time for survey design is that you need to have some randomization. That is, your sampling, your choosing of your sample should be random with respect to your question. So randomization, replication, you need to have multiple points, multiple areas that you get information from, and control of variation. Why do you want these? Randomization so that your sample is representative of the population. Replication you basically want inference from multiple independent samples, right? Multiple samples which are independent of each other. So in that entire area, going to multiple different locations. And control of variations because you want some precision in your parameters. You want to say something with a little bit less uncertainty. When you go over there, you're like, I'm not 100% sure. When you come out from there, you don't want to be like, I'm kind of like, I'm not 95% sure types. You want to have some little bit of precision and a little bit of confidence in what you come out. So just going into this a little bit more in detail, randomization, again, you want to ensure that the sample is representative of the entire population. And essentially that means that you need to, what does this mean? It means that your sampling units are chosen randomly from your population, randomly with respect to your question. Your replication means you select multiple independent sampling units and you select multiple sampling units. That is what you refer to as your sample size, and it has to be independent of each other. Won't get into why it has to be independent of each other, but it has to be independent of each other. That's because you're getting like multiple pieces of information. That's what you're doing. You're taking multiple pieces of information from the same area. You need to make sure that you avoid pseudo replication. For instance, if you're looking at your rodents and saying what is the whether the rodents are sick or not. And then you capture that same one rodent like 5 times and say, oh, it's sick, it's sick, it's sick, it's sick. Then that is called pseudo-replication. Because these are not independent, separate data points that are coming from your population. And to control your variation, you can increase sample size, but there are also some things that are leading to that variation, right? And some of those things you might be interested in. There may be deer present in one area, not present in an area because of the water. You might be interested in that. So you take that into account as well. Yeah.
Divya, could you please elaborate a little bit about the pseudo-replication? So did you mention that it's about the rodent, like an animal you sampled multiple times? Okay. Yeah. So in that case, let's say the sampling site, how a sampling site can be replicated as a pseudo-replicate.
Okay. So suppose you're looking at fragmentation, right? You're looking at fragment size. So you say that forest size is going to lead to lesser number of species. And you have 10 different, say, you have like multiple fragments of different size. And your question is, does a smaller fragment have lesser number of species? Which means that you need to have multiple fragments of smaller size, multiple fragments of medium size, multiple fragments of big size. So if you have the same fragment of a smaller size that you go and repeat visit, that might be pseudo-replication. Yeah. It depends on what your question is, but as phrased, that might construe, and as if you analyze it directly at the inference of that fragment, that might construe pseudo-replication. You had a question? Yeah.
So I was just detection probability. For example, the species
detection probability
is there.
is coming. We'll talk about it a little bit later.
Okay. Okay. Yeah.
Did you have detection probability issue with respect to this?
Yeah. So yeah, it's present there, but you didn't detect. Yeah. That's
Yeah. That's exactly what it is. We'll come to it. We'll just talk about it in a little bit. Might be just before lunch. Lunch is there, but we're not able to see it types.
See, I have another question.
So how do we, you have a question.
Yes. So I have a doubt. If there is a boundary differentiation between the same species, like one clan is not crossing the boundary with other clan, while sampling you differentiate as a different samples or the same one?
You mean different herds?
Yes, different herds.
That again depends on what your question is.
Okay.
Yeah, if you're looking at, for instance, I think Varun mentioned line transects, right? So you might want to count these animals. You might want to go walk along a transect, and you might want to see, okay, how many deer are there? And you see one deer and you count it as one deer, and then you see, oh, that's not one deer, that's like 7 different deer. So now the detection of all of those other deer have been influenced by your detection of that first deer. You're seeing those other 7 because you've seen the first one. You see what I'm saying? So you need to count them as, you basically need to take into account that you've seen them together. They may be 7, how did I say 7 or 8? They may be 8 different animals, but you have seen them together. Yeah? So if your inference is how many deer are there, it will still be that, that 8 deer will go into your count. But you have seen them together. That will go into your observational process. Yeah? All right, so there are a bunch of different really simple ways that you can ensure that you can have randomization. You can select multiple different independent samples. And I'm going to talk about, have some of you at least heard of these methods? Have all of you heard about these methods? How many of you have? How many of you have heard of simple random sampling or have implemented simple random sampling? Noah. Okay, thank you. Some of you have? Okay, all right. So simple random sampling is basically really simple. You have 100, that's in the name. You have 100 different units or points over there. You want to choose 10, you choose 10 randomly. It's like pulling marbles out of a sock, you just choose 10 randomly. Without any consideration of anything, no consideration of what is forest, what is grassland, what is urban area, you're just randomly choosing it. So you have n, small n units, that's your sample size, out of large n, which is the overall number of things that are there in the entire population, selected such that every unit has the equal and same probability of being selected. So if you are selecting the deer, for instance, maybe you want to take like blood samples from them. There are 100 deer in the forest, you want to select 10, you select them randomly. If you're selecting locations, it can apply to locations in space, it can apply to animals in a population, Can apply to like the households around which you are sampling. Can apply to the caves within which you are sampling the bats. It can apply to any of that. It's really simple to implement, but if you want to take an additional step, maybe ensure coverage of all areas, maybe ensure like that some underrepresented groups are not covered, then this may not be the ideal way. But it's really simple, really easy to represent. This sort of the landscape that you might consider. You may not want to climb up and down those huge cliffs, but simple random sampling is not going to help you with that. You might get a point up there. You might get a point up there. What do you notice? Some of them might be closer together. Some of them might be further away from each other. That's part of the random process. You may think that, oh shucks, there's a hole over here. What do I do? That's part of the random process. You can't take this one and plug it over there. Then you've messed up your simple random sampling. But suppose you think, oh, I don't want these to be there, right? Then you can use a different method, which is called systematic sampling with a random start. So systematic sampling with a random start is just that you're going to start at the beginning randomly and then place your points every so much meters apart. Maybe every 100 meters apart, maybe every 1 kilometer apart, and so on and so forth. I'll just show you what it looks like. It's like this. So now you can see all of your quadrats are placed sort of evenly. You might have many deer over here. You might have very few over here, or rodents, or whatever it is. Doesn't matter. This is part of the process. That is what ensures that you're representing the entire system. You still have those points on the cliff, but that's part of the scene. So you randomize your first sampling unit, and then you take a non-random sample of the rest by placing them every 1 kilometer or so apart. The assumption is that there is no pattern that you're capturing over here. Say you have groups, right, maybe, and your groups of animals, maybe primate groups. There are langurs across this entire area, and the langurs for some reason are 500 meters apart. For some reason, they're 500 meters apart. Or maybe there are hills, or there are like rivers. And those rivers are placed like, are there in the system, and they are like some 500 meters apart. And it just so happens, when you select, your groups kind of follow, your grid cells kind of follow that same pattern. That's an issue. You need to make sure that there is nothing which is going to influence your parameter of interest, which is following the same pattern as your separation. For the most, it won't. I mean, if you think about it a little bit, it won't.
Why would that make an issue? Probably you are getting an actual, suppose the same example. So your pattern is following the same dynamics within the system. That would give you approximately accurate quantification.
It may, but if that pattern is influencing what you're going to study, then it might give you misleading patterns. I'm trying to think of something which might work in this particular case. But if, for instance, say you don't have like so many rows, no, it's difficult to think with so many rows. But say you have like some sort of a contour line. Say you have like sand dunes. You're looking at it in sand dunes, and you're going up and down, and you only capture like the top, and you don't capture the bottom. Maybe as some example, off the top of my head. You had a question. I said for the simple random sampling. I'm repeating it because the. If you shift it, if you say that, oh, there's nothing over here, but I really want to go over here, and you say there are so many over here, I'm going to shift this and put it over there. Because this is your random selection. When you shift it and put it over there, there's some subconscious thought process in your mind that is pulling it out there. When you're saying simple random process, it's not you putting, yeah, go ahead.
So I'm sorry, I know we're running late and I probably shouldn't be talking, but I think that when we think about doing a science or research, we are always thinking about trying to maximize what we get. I mean, subconsciously, we're like, how do I work such that, I get the most data, the best results, and I kind of know that, these animals are there, or if I put my traps here, like, how do I live with 3% trapping success? It's depressing, right? But on the other hand, we cannot be biased. If we are biased, then we are not going to get, that's wrong. That's incorrect science. Does that make sense?
You had something to add?
Just one point to add over here is that all of these different designs that we're talking about, this is to do with what she was saying earlier, which is that you can't go to every single inch of your study area. So based on where you are collecting data, you want to be able to extrapolate to areas that you haven't gone to. So if you want to say, okay, I've got, whatever counts over here, and based on this, I'm going to say, okay, for areas that I haven't been, that this is the kind of expectation for that area. In those kind of cases, you need to have random sampling, stratified sampling with a random start, all of that. If you are not trying to make inferences in space for places that you haven't been to, then your design might be slightly different, where you're maybe trying to maximize your capture of certain animals. So you're putting camera traps, you're putting Sherman traps, you're putting tomahawk traps in locations where you'll have the maximum chance of that animal getting caught. Because here you are interested in those individuals or that community, but you're not saying, okay, I haven't put traps over there. So based on the density I've got over here, I'm going to say that there are that many animals there as well. If you're not doing that, then this randomization is not as relevant. It's in this kind of where you're doing spatial sampling, or you're basically making extrapolations in space to other places.
I also want to say one more thing. Like sometimes people say, oh, I just randomly selected. But they actually mean that I went over there, looked at a location, and I chose a point and random. That's not random selection. It's generated from it's generated beforehand by, and I'll just talk about it in a little bit, but you can implement these in most of your QGIS systems or any kind of software systems. So when you say random generation of, like say you have 100 points and you're randomly selecting them, you're doing it in R, for instance. You're saying like select sample 10 points out of 100 in R, or doing it in Excel or something like that. R is better than Excel, but you're doing it in R or something like that. Or if you're selecting points in space, you do it in QGIS. It's not that you are going to that location and standing maybe in one place and say, okay, I'll select that point. Because even if you're not consciously doing it, you're subconsciously, there's some thought process in your mind that goes into selecting that particular spot. Now, if, like Varun said, there's some reason why you want to maximize, once you get to a point, you might want to maximize. So there are certain conditions under which you can go to that point, and then maybe if you're putting out mist nets like how Umesh does, you might put it in a specific location where it would be suitable for the mist net, or you might increase your capture of the birds. So that amount is fine. But if you say that, oh, I only want to walk, you remember like that guy along that road, and choose random locations along the road and just say, okay, I'm going to stop over here and then I'm going to stop over there. That is actually not random. You wanted to say something? Oh, you're just standing. Right, so it can provide, we were talking about systematic sampling, it can provide a better sort of spatial coverage, and it maintains some level of spacing of samples. Now this is important because say you want to keep some sort of criterion saying that all of my points need to be separated by at least 500 meters. Then this allows you to do that. That allows you to say maintain independence of your sample. In some particular cases, you might also want to stratify your samples. There may be something where you're interested in looking at low, medium, high, or you might be interested in looking at close to villages, medium area from villages, far away from villages. Or you might be interested, who is interested in the deer-human interactions? So you might be interested in interactions where there is agricultural fields and where there are no agricultural fields, or something like that. There's some particular strata or some particular groups. Or you're thinking about rodents, you might want to have different species, or you might want to have the small ones and then the big, like small rodents and big rodents, or things like that. So there may be certain groups or certain strata, certain areas in your sampling units itself that you might want to, a priori, you're interested in looking at, and you want to sample within each of those. So you want to make sure that you have 10 samples of these, 10 samples from here, 10 samples from there. Say, I'm just going to show this now itself. Say you have 3 types of forest, and you want to ensure that you have at least 10 samples in deciduous forest, 10 samples in semi-evergreen, and 10 samples in evergreen. So that is when you'd use a stratified random sampling. All you do in this case, there's only one additional process. You define your strata and then you do a simple random sampling within each strata. You might, yeah, so things, I guess that kind of covered everything, right? So you may think of water holes where you see more animals and away from water holes where you see less animals. You might want to look at their roosting spots, for instance, where you expect them to be there in the evening time. And you want to look at what is happening in each of these different places. You might want to then choose locations in each of these places and then get data from each of these places. That allows you, importantly, to compare across these strata. So you usually do this if you have underrepresented groups. You may have one particular land use that is very underrepresented, may not really come up when you do a systematic or a simple random sampling, but you want to make sure that you have enough samples over there. You might really love these cliffs and it might be really, really important for your particular species, but it may not really come up. So you might want to force it out and say that I would definitely want to get 10 samples from this area. Yeah? So in that case, you would do a stratified random sampling. Usually you predetermine what proportion or what number of samples you have in each strata. You choose your strata and then you just do a simple random sampling within each strata. This ensures representation, which means enough sample size to make some inference about each of these strata. And it is really useful when you want to compare across groups. Now there can be the flip of that also. You might have clusters. You might have like multiple locations where you have slightly different types of things. For instance, you may have near mid distance from village sites, but you're looking at different villages. So with the rodent sampling, sorry, I'm taking like the examples of which had already been given. For the rodent sampling, for instance, you might be interested in really close to the house, in the grain site, and in the paddy field. And you might want to do that for 10 different villages. So you now have in each cluster, for each village, you have these three different points which represent different things. And they form a cluster and you have 10 different clusters. So the villages have been chosen randomly, maybe the houses have been chosen randomly, and then you have these three different variations within each place. So that is also useful when you want to compare those variations across places. All right, so a couple of additional things and then I think we'll have to stop. Covariates are really important and important to consider when you do your sampling itself. So when you are thinking about why are we doing this stratified sampling? We're doing it because we want to compare that evergreen versus whatever, right? Why are we doing the cluster sampling? Because we want to see whether there is more chance of getting the rodents maybe next to the granaries versus next to the houses versus next to the paddy fields, or whether there are different species in these areas. So there's something which is there of interest. You might want to, going back to where we started, we're thinking about like the number of tigers or tiger presence and prey presence. So we want to make sure that we capture areas which have low prey and capture areas which have high prey. Otherwise, there's nothing we can say about any of these things. Or percentage of forest. We want to capture areas which have low forest and capture areas which have high forest. Otherwise, we can't really say anything about it. So thinking about covariates from the beginning itself, going back to thinking about your models from the beginning itself is really important for your study design itself, because that will determine to an extent where you can also place your points. And it can ensure that you can answer those questions at the end. Yeah, so these are just examples in forest versus agricultural fields, near versus far away from caves. And I think I'll just say this bit and then after that we can stop for a bit. But the other thing that we might want to do is purposive sampling, which I think is what some of you have done over here. So purposive sampling is when you're not really interested, like Varun said, in extrapolating to the entire population. You want to maximize your chance of getting the animal. Or you might have randomly chosen a sampling location, and in that sampling location, you want to maximize your chance of getting the animal. For instance, you choose a sampling location for camera traps and you choose a random direction, and then that camera trap is facing like a tree trunk. You're not going to get anything, right? Like that's a waste of time and camera trap and people and all of that. So in some cases, within some constraints, you want to maximize your chance of detecting the species. So this might be about the spatial locations where animals frequent. So within a particular space, you might say, in a 100-meter radius around the point, I'm going to choose either trail junctions or water holes where like the animals there may be more chance of seeing the animals. Or you may say that I'm going to make sure that I'm next to caves. So I'm going to go to caves such that I maximize my chance of seeing bats, things like that. Or if you're looking at human interactions, then obviously you want to go to areas where people are there. If people are not there, there's no question of You might, in that case, also want to have baited traps. So Varun spoke about Varun spoke about traps, live traps, and camera traps. When you don't use baits, it just captures whatever is there or happens to come there, right? So if you want to know what is there in that location or what happens to come there in that location, you can't bait. But if you want to put those traps out there and make sure, maximize your chance of capturing that animal, then you might want to put baits. So this can happen with camera traps, hair snag traps, live traps, and very, very important for disease ecology, where you might want to go to a location, but within a larger area, you want to maximize the chance of getting the animal. You can put food, you can put olfactory baits, CK1, very, very useful for cats. Raccoons, Varun spoke about like stuff that you put over there. I think Arjun made a whole lot of peanut butter in to put for rodents in our, like either smell, pheromone, anything like that. And this is, I guess, the last thing that I'll say, which is, so just to recap, we spoke about various different types of sampling that we can do. These mostly deal with the spatial sampling process, but the same thing can be applied to animals as well, or units, other units as well. Just to keep in mind, sometimes there are natural units, like for instance, ponds, caves, those are all natural units. Sometimes we have to impose units. For instance, we might impose a grid network on this entire area and then choose grid cells randomly, yeah. And all of these, like mentioned, can be implemented via most GIS software or R, and it doesn't, so randomly choosing this is not, okay, I'm going to choose this, 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 and this, because subconsciously you're like, okay, I want to go there, and that comes into play. It means that you're doing it, implementing it via some of these systems. So that's about it. I'll stop over here, and then we'll talk about detection process after lunch.
So much. There was tour de force, and we'll have more in the afternoon. We'll take a break for lunch. Thanks, Divya. I didn't introduce Divya. Divya is also an exceptional statistical ecologist and also works on primates and connectivity. Connectivity.